Hello and welcome, everyone, to today's session of Global IMMUNOTALKS. And this is the seminar series that brings all aspects of immunology to a global audience. So today I have the privilege of welcoming Jenny Ting. Jenny is professor and director of the for Translational Immunology at the University of North Carolina at Chapel Hill. And she has had a long and distinguished research career spanning a vast number of aspects of immunology, from cancer to autoimmunity, autoinflammation, infection, and work on the microbiota. And Jenny's work has ranged from the transcriptional regulation of MHC2 expression, which in fact, I think her interest in C2TA, if I remember correctly, led her to the discovery of the human NLR gene family. But she has also worked on the role of MHC and TNF family members in neuroinflammation. And she has had a long history of many prestigious recognitions. Among others, she's been elected to the National Academy of Sciences in the US, as well as the American Academy of Arts and Sciences. And she was also president of the American Association of Immunologists from 2020 to 2021, and has served on a number of different boards, including the NIAID Board of Scientific Council, the advisory board of the American Asthma Foundation, and she's currently chair of the board of directors at the Burroughs Wellcome Fund. Now, in a minute, I'm going to ask Jenny to deliver her lecture, but as is usual in the series, we always start by asking our speaker a personal question before the talk. And the question, Jenny, is in what ways has interdisciplinary collaboration enriched your scientific perspective?
Thank you so much for that. Such a kind introduction. It's great to see you virtually. And I just want to thank everybody for joining in. Yeah, in terms of our research, I've been thinking about it. We got this question. I was late in picking a question, but a few minutes ago. I think my talk will illustrate how important it is for collaboration. Nowadays, even way back, it's really hard to single-handedly carry on all the research and research directions in the lab. So we have so many collaborators. And when I go to meetings or seminars, give seminars, one of the things I enjoy the most is really connecting with other people and forming collaborations. We have several collaborations, for example, with people with completely different disciplines, like nanotechnology and, a lot of protein structure people. And I don't claim to be a protein structure person by any means, but because of what they've contributed, we were able to do a lot of really nice research. So I think collaboration is really critical. Being a good collaborator is a key to that. So we have been very, very lucky finding so many great collaborators.
Yes, and of course, immunology is increasingly interdisciplinary and touches so many aspects of biomedical research that it's easy in a way for us to collaborate with many others. So thank you, Jenny, for those words. And so now I will invite you to share your screen and deliver your talk, which is entitled The Importance of Innate Immune Receptors in Innate and Adaptive Immunity. So Jenny Ting.
Yeah, so thank you again for that very kind introduction. So what I will tell you today is two different large vignettes. One is the role of innate immunity or innate immune receptors and adaptive immunity, and it'll be a pretty short section. And then the second part is what are we doing in terms of trying to study human immunology and the role of innate immune receptor in those systems. So I have used this slide for decades now, sorry. And that is all the innate immune receptors and sensors. There are many others that are not in this figure, including scavenger, for example, surfactant receptors. But we all know that there are key membrane-associated receptors that detect innate immune disturbance, either from infectious diseases or cell damages. But many of the innate immune receptors are actually inside the cell. So that is really unique to our discipline, in that when you think about receptors, most people don't think about receptors inside the cell. So what I will tell you today is I will mention certain DNA sensors such as AIM2, CGAS, and I will also focus on this family of proteins that Kadana has already introduced to you, the NLR family of proteins. Now, this is a very large family comprised of 20-some members in humans and 30-some members in mice. And like he kindly introduced, we started this because we found homologies between C2TA and this large, at that time, undefined family, although several members were already known at the time we published the family. And these family members are pretty amazing in that they're preserved from plants, and there are hundreds to thousands of plant disease-resistant proteins that share homologies of the NLR or nucleotide binding domain and the leucine-rich repeat sequence. And this is also found in plants and more recently in prokaryotes. Now, one of the key functions of NLR protein is an inflammasome, and I'm sure almost everybody is familiar with this function, and that's in the production of caspase-1. And in general, these are the key inflammasome NLRs. Some are better studied than others. For example, NLRP3 is the best studied, and it associates with a common adapter protein called ASC, and you'll hear me mention this. There's lots of beautiful structures that are done in our field, and NLRP3 associated with ASC activates procaspase-1, which then becomes caspase-1 to digest as downstream signals, including pro-IL-1, IL-18, and gasdermin D. There are others as well, including NLRP1, which is really a hot topic right now, NLRC4, NLRP12, NLRP6. Some are, like I said, better studied than others. And one of the remarkable things is that NLRP3, for example, is associated with several autoinflammatory syndrome indicated here, first depicted by Hal Hoffman. Now, although NLRs are thought about as mostly an innate immune system, what we have found is that many of these are important in the adaptive immune system. So this is just an example, a list of NLRs that are important in the adaptive immune system. For example, NLRC3 is well expressed by T cells, as well as by B cells and NK cells. And we published quite a few years ago, two of the postdocs in my lab, who are now both back in Japan, showed that NLRC3 can prevent K63 activation of TRAF6, therefore blocking NF-kappa B activation, therefore blocking all these downstream signals important for T cell activation. In collaboration with Jason Whitmire's lab at UNC, we showed that this NLRC3 prevents an overzealous CD4 T cell response during LCMV infection as well as in EAE. And another protein in RX1 shown by Haitao Guo that it associates with a important protein that regulates oxidative phosphorylation and therefore altering immune metabolism that favors HIV activation. This is shown here, also in Nature Immunology. And finally, the example of AIM2, which I will spend a little bit more time discussing this. And this is work in collaboration with Yisong Wang and Zhengli Guo in his lab and done by Emily Chow in our lab. And AIM2 is one of these inflammasomes that's not an NLR protein, and it was discovered by these three groups listed here in 2009. And AIM2 is one that's where the ligand is very well defined. It's a double-strand DNA that binds to AIM2 and causes association of ASC and procaspase-1. And Sam Shell and Kate Fitzgerald has done beautiful work on the structure, and what it does is, again, activates caspase-1 upon DNA binding, leading to processing of IL-1, IL-18, and gasdermin D. So this is always thought of as an inflammasome, well-defined DNA sensor. And so I would just tell you that this is also actually well expressed in T cells, especially in human T cells, and which led to this work. So years ago, we were looking at the disease EAE. This is a model for multiple sclerosis. It is T cell mediated, and many people have done a lot of work in this field. And what we were asking is, what about the role of inflammasome in the EAE model? And what we found is when we have an NLRP3 knockout, the disease is severely attenuated. So NLRP3 is a pathogenic factor in EAE, and this paper is published by Dennis. And subsequently, several labs, including Marie Shinohara's lab, showed that if you look at ASC as well as NLRP3, they both have very reduced EAE score, and the caspase-1 inhibitor can inhibit this process. So we repeated the additional ASC and caspase-1, I'm sorry, here, and this is the data where when we're looking at caspase 1 and 11 knockout as well as ASC knockout in the EAE model, there's hardly any clinical score. So this is a very, very robust system where knocking out inflammasome caused the reduction of EAE. So we wanted to know about the role of AIM2. Actually, this was asked by a reviewer in a grant. So we put this knockout in, and lo and behold, what we found is instead of getting a reduction of disease, such as these other inflammasome knockout, we have actually an enhancement of disease compared to these wild type. So this is work done by Emily, who later worked with Jing Li. And she kept on showing the same data, and I kept on not believing it until she get a large number of mice from about 9 experiments. And finally I said, oh, I think this is true. We should probably look at this in more detail. And she showed that indeed in AIM2 knockout mice, even though we got rid of the inflammasome, we have increased demyelination, increased inflammation, but no changes in the spinal cord for IL-1 or IL-18. So a number of studies led us to realize it's not the myeloid cells that are affected in this disease, but rather it's AIM2 and T regulatory cells. And we did cell transfer, et cetera, to delineate that. We also saw that AIM2 knockouts has reduced IL-10 expression as well as Treg numbers. So this led us to Tregs, and we showed AIM2 is expressed well in T cells, including Tregs. So we made a T cell specific, Treg specific knockout. We're using the FOXP3 promoter, and by doing that, we showed that deletion of AIM2, excuse me, only in Treg cells resulted in increased clinical disease, showing that AIM2 in Treg has a function that is promoting T regulatory cell function. And if we look at Tregs without AIM2 indicated here, what we found is we actually have reduced Treg numbers, but we have increased of interferon gamma expressing cells. So we had no clue at that point what is the pathway. So we did RNA-seq analysis of T regulatory cells, both inducible as well as native. And what the data were the same, and I just wanted to show you one of the two sets of data. And what we showed is that in the knockouts, we have increased mTOR, increased PI3 kinase, increased glycolysis and MYC. And at that point, we started to think about immune metabolism, and which was introduced to our lab by Elena Rampinelli. And so what we did is we look at all the immune metabolic biomarkers, and what we found is that AIM2 knockout Tregs have increased here glycolysis, but decreased the AIM2 knockouts have very reduced fatty acid oxidation. So this is, again, immunometabolism that's shown by others to be linked to a Treg immune metabolic phenotype. So we wanted to know, what is AIM2 doing to cause this? We would imagine it's not with the inflammasome. And so we did an IP immunoprecipitation mass spec to identify AIM2 with the lab uh, um, with Sean Chen's lab at UNC. And so this is done with primary T regulatory cells. So this is pretty painful. We use wild type and AIM2 knockouts and activate it in the presence of TGF beta to get T regulatory cells. And after 24 hours, immunoprecipitated with anti-AIM2 antibody. And then the tryptic digestion to get mass spec. And we looked at the proteins that were pulled down. And out of these, we could link it to the PI3 kinase and AKT pathway was RAC1. So RAC1 is known to recruit this phosphatase called PP2A, and this causes dephosphorylation AKT. And I forgot to mention that in our phenotype, one of the strongest is in AIM2 knockout, we see this phenotype of increased AKT phosphorylation in AIM2 knockout. So we wanted to see how these two are linked. So we took wild-type Tregs and AIM2 knockout Tregs, and we overexpress RAC1 and PP2A in these Tregs. And in the wild-type Tregs, what we found is we can actually reduce PAKT phosphorylation if we put in these two, which is known to dephosphorylate, AKT. But in AIM2 knockouts, we don't see this, suggesting that this step is dependent on AIM2, that all three, and then we also showed co-precipitations of all three together. So when we go in vivo into the spinal cord of EAE mice, what we found is that in the AIM2 knockout and T regulatory cells, these cells indeed have increased PAKT, which would be antagonistic to Tregs. So just to conclude for this part, what we found is a different function for AIM2 that's non-canonical. It's not an inflammasome induction, and this is where AIM2 can recruit RAC1, which can recruit this phosphatase to dephosphorylate AKT, and therefore disallowing the progression of PAKT necessary for other T cells, but rather this leads to T regulatory cell stability, which we showed also in the paper. So in addition to T cells, I just want to show you two slides where we also looked at B cells and other innate immune receptors. And this is looking at the Ting pathway, and this is well known, I'm sure, to everybody, where the discovery of James Chen, where DNA from different sources can activate CGAS, and this causes the activation, the production synthesis of CGAMP, and then it activates CGAMP as an agonist of STING, and when STING is activated, what you have is TBK1 and FKB B activation, interferon, inflammatory cytokines, etc. And this has been of great interest to cancer immunology because it's shown by several people that STING has two effects. One is on the interferon in cancer cells, and one is on myeloid cells, such as dendritic cells, macrophages, etc. So you have two activation in the tumor microenvironment, therefore causing better immune phenotypes that could lead to increased class 1, class 2, CD40, CD80, etc. through these type 1 interferons. However, there's lots of counter problems as well in that there's other studies that show that this pathway can, for example, induce T regulatory cells, and monotherapy for STING in cancer has been disappointing. And most mouse models require intratumoral injection. So I just want to show you one slide of our linking of this pathway to B cells in a very surprising link. And this is done by Cici Liu in our lab, who is now a faculty in China, together with Yulia Payaleva Gupta at UNC with her postdoc, Rahul. And so what we found is that B cells, surprisingly, when they're activated by STING, when they encounter STING systemically. So in our system, we were looking at tumors and STING injection, not in the tumor, but systemically. What we found is B cells are activated in the spleen. They become B regulatory cells. They go to the tumor environment. They secrete IL-35. They also secrete IL-10. Both of these are immunosuppressive, and they stop NK cells from proliferating, and these NK cells, therefore, cannot uh, eliminate the tumor. So what we found is if we have a STING activation, but we block this whole pathway by reducing IL-35 with anti-IL-35, we can turn 5 different cold tumors into hot tumors. So this is another unexpected finding that B cells can be activated by STING to become B regulatory cell. There was a previous paper showing that B cells can respond to STING. So this pathway has been known, but we showed that this is in B regulatory cell, and this was published a couple of years ago. So the rest of the time, I really want to devote to innate immune receptors and innate immunity, and just telling you some of our studies in human and how we're trying to transport knowledge in the mice into a human system. And some we are using humanized mice. And the first example I'll give you is really genetically humanized mice in diseases. And this is our collaboration, long-term collaboration with our wonderful collaborator, Bev Kohler, who deserves the really majority of the credit for what I'm going to present. And Bev is the person who made the first beta-2 microglobulin. I think there were two. She's one of the two authors on one of the beta-2 microglobulin papers. And also she's made tremendous really contribution to the cystic fibrosis field. And so what Bev has done is made these, instead of single mutations of human mutations into the mouse gene, she made synthetic replacement of mouse genes with the human counterpart. So this is really difficult, but because you're putting very large sequences, including the 5' regulatory sequences, intron, exons, 3' flanking sequences from human into a mouse that doesn't have the gene anymore. And these are two of the papers. They're both on NRP3. Like I mentioned, this is a protein very important in arthritis because it causes several autoinflammatory diseases. And that's extensively studied, as I mentioned, by Hoffman, as well as by Dan Kastner and his wonderful colleagues at NIH. So I will just only show you two slides from those two papers. One is the synthetic replacement of mouse NRP3 gene with its human counterpart, normal counterpart, or the disease-associated counterpart. And this one replicates the human disease. And so what she did is took out NRP3 from the mice and replaced it with the human NRP3 allele. And then she also replaced it with the disease-associated allele. And what she found is in the latter, spontaneously, these mice have osteoporosis as well as arthropathy shown here, and many of an increased response to LPS. And why is this important? Because in the mouse system, where two labs have made the very important initial discovery of inserting the human mutation into mice, into the mouse gene, they were able to show increased autoinflammatory disease. But infrequently, the mice have very severe outcome, including death. And we know these diseases in humans do not really result in such severe death in the case of cold-induced autoinflammatory disease. And ours better replicate in these because these mice can breathe fine. The problems they have are mostly in arthropathy. So this better, we think, mimics the human disease. More recently, we have also made the human promoter and human allele. So this is a proper synthetic replacement. In this case, the human promoter was replaced with the mouse promoter. And interestingly enough, the mouse promoter, when we use a typical inflammasome activator, ATP and LPS, what we found is the one with the mouse promoter, they both had the human allele, produce better amounts of IL-1 and better activation of the inflammasome. And then just showing mouse to human differences, we also look at the human mouse strain, a humanized mouse strain with human disease, NRP3 disease allele. But here you have human promoter and mouse promoter. And this one, which has the human promoter with the human disease allele, it has spontaneous meningitis. So in this case, the microglia are activated And there's meningitis associated with it. And this is interesting because in the human disease, there's also a component that's CNS related. But if we use the mouse promoter, we don't see this, suggesting under different stimulus, the mouse and human promoter can actually work differently. So these experiments underscore the importance of looking at human either promoter or allele and having genetically replacement of mouse genes with human genes. And several labs have other approaches to look at humanized mice. The second one I will tell you about is really using cultures that are from human tissues. So in our case, we're using 3D air-liquid interface cultures of the lung, and this is really done with Scott Randell at our institute. And this is looking at SARS-CoV-2. And so when SARS-CoV-2 first came, we were interested in what causes the inflammasome. There are many labs that have done really nice work addressing this question. But our question was really wanting to know, what if we look at the airway epithelial culture, because those are the cells that are infected. What happens with those? And what happens when those are incubated in co-culture system with your immune cells? What happens when we have immune cells? So we feel like that may better replicate what we see in the infected lung. And this is just a paper from Ralph Barrick, a colleague at UNC, really an eminent SARS-CoV-2 scholar who showed with Scott Randell that a GFP expressing SARS-CoV-2 virus can infect these cultures that are done with air-liquid interface. And these are all human epithelial cultures from human donors. So these are primary cultures. They grow with limited passages. So they are kind of pretty difficult in some ways to work with. They're not like a cell line for sure. And so this shows that these cells can be well infected with SARS-CoV-2. So Kat Barnett in our lab, who will be going to the University of Michigan as a faculty, asked the question, what is the event that happens after cells are infected? What she showed is after cells are infected in the airway epithelial, the SARS-CoV-2 virus does not cause inflammasome activation in these cells because other people have shown there's an inhibitory pathway that inhibits this process. Instead, these cells die, release RNA, release double-strand DNA, and this forms as a signal for inflammasome. Inflammasome requires two signals, and these dead DNA provides signal 2. In terms of signal 1, SARS-CoV-2 produces the E protein, which provides a TLR2 activation, which was shown by Tirumala Kanaganti to be important for SARS-CoV-2, and that activates IL-1 production. And so you have two signals. One is from the virus itself, one is from these dead cells, and the two signals together cause inflammasome activation in these myeloid cells, causing IL-1 release. Then we showed IL-1 release can go back and cause IL-6 production, tremendous amount of IL-6 production in these epithelial cells. So this depicts a much more complicated system where you have two cells interacting with each other, amplifying IL-1 production and IL-6 production, and both of these are critical in SARS-CoV-2, including in long COVID. But as I mentioned, during our observation is SARS-CoV-2 infection, and this is observed early on from the first postdoc who started the SARS-CoV-2 work in the lab, Dinka Song, who is now in China as a faculty, and we showed that there's lots of cell death, but many people, again, have looked at cell death in this system. And the question we wanted to ask is, what is the event? What is the cell death event that happens in the primary cells that are being infected? Again, those are airway epithelial cells. So there are several pathways of cell death. There's actually quite a few, and we decided to just focus on 3: apoptosis, pyroptosis, and necroptosis. And just to show you the markers we use for apoptosis, we use caspase-3, and some of the studies we unpublished, we've used caspase-7, and pyroptosis, we use Gasdermin D processing, and for necroptosis, we use mostly MLKL, but we also looked at RIP kinase 1, kinase 3, but MLKL is more specific for necroptosis. So during SARS-CoV-2 infection of epithelial cells, these are airway epithelial cells. We also did it in A549, and the results are fairly similar. And what we found is during infection, we see increase of SARS-CoV-2 M protein, and this is done by Kai-Sheng Liang, who's now a graduate student in the lab, who's now a postdoc in Richard Flavell's lab, and also by Kat Barnett. They're co-first authors on this paper. And what we showed is LDH also goes up with time. And if we look at these different markers, we can see Rib kinase 1, Rib kinase 3 phosphorylation. So you should look at the phosphorylated, the P lanes, and they're phosphorylated. The level of expression also goes up with SARS-CoV-2 infection. This is time of infection. And most important, MLKL phosphorylation goes up. So this is shown here. And then if we look at SARS-CoV-2 infection in terms of apoptosis marker, they actually lag behind MLKL activation, for example, which happens pretty clearly at 48 hours, but we don't see this until 72 hours. So we start to see caspase-3 processing over here, indication of apoptosis. In gasdermin E, also at pyroptosis, we see this later. This is expected because this can be a consequence of caspase-3 activation. But SARS-CoV-2 virus M protein, we can see it all the way from the very early time point. So I should say this is done at the BSL-3 facility at Duke, and we really treasure our collaboration with all our colleagues at Duke. We looked downstream and we looked at gasdermin D expression in these HAE cells. Not surprisingly, we don't see much because gasdermin D is not well expressed in these cells, and we don't see much IL-1 expression either. This is a positive control for IL-1. And when we look at another indicator of inflammasome activation, ASC, we don't see much either. So this suggests that two events are happening that we measured. One is necroptosis, one is apoptosis. So the question becomes, what happens in the cells when they're infected with the virus? Which cell death process happens, or multiple cell death processes happen? And so we address this question. So these are either airway cells or A549. I think all of these are airway cells, and these are the primary airway cells I was telling you about. And so we stained for phosphorylated MLKL, look at those that have M proteins. Then we counted, not we, but Kai-Shin and Kat did the laborious work on counting cells. In some experiments, it's thousands of cells, and other experiments with the airway, it's much more difficult because they're multi-layered. So these are hundreds of cells that we counted. And what they found is that the cells that have M protein, that is, they are directly having the SARS-CoV-2 in the cell, these are primarily MLKL positive. There are very few cells that are MLKL positive that don't have the virus. This is in contrast to when we look at caspase-3, which is a marker for cell death. And here, we don't really differentiate. Many of these cells seem to have some caspase-3 and M-positive cells, and some that are M-negative, they still have caspase-3. So Kai-Shin and Kat designed an experiment, which is do limiting dilution of the virus. So MOI is increased in a dose-dependent fashion. And the question they're asking is, at the very low dose of MOI of 0.01, we would expect that the cells that are expressing N, which have the virus in it, that they are infected. And they are not some bystander cells. So even if you dilute down the virus, the M-positive cells are still infected. And we look at those cells, and those all have MLKL, indicating that these cells are undergoing necroptosis, the process of cell death. In contrast, when we look at caspase-3 positive cells that have M-positive markers, we looked at the very low dilution. And here, none of the M-positive cells, I should say all of the caspase-3 positive cells don't have the virus in them, suggesting that this is a bystander defect. And this is also shown at MOI of 0.1. And here at the higher MOI, we start to see both. And so we're suggesting that bystanders are a major component of the cells undergoing apoptosis during this infection. They do not have the virus in them. So to address this even more, we look for cells that are double stained for MLKL and also for caspase-3. And this is for phosphorylated MLKL, and this is for cleaved caspase-3. And what we found is upon infection with the virus, we're looking at the percent of cells with MLKL and caspase-3 not negative. And all the MLKL cells that we can find do not have caspase-3. There's a tiny little amounts that are caspase-3 positive indicated here, but almost nothing, none of these cells have both, which is this one, unfortunately. I'm sorry, almost all the cells are most of the cells, I'm sorry, don't have either of the markers. And the reason for this is we believe most of the dead cells have floated away. So these are the live cells that are remaining. But if we look at cells that have double markers of both necroptosis and apoptosis indicated here, we almost saw none of the cells would have these double markers, suggesting they die either by this pathway or this pathway. So we next knock out the MLKL gene, and this is indicated here. And we did 3 different experiments. And when we look at caspase-3 in these knockouts, statistically, there's no difference between wild type and MLKL knockouts, suggesting that these 2 processes are independent of each other in the airway epithelial cells. So then one wanted to know if this happens also in COVID-19 autopsy lung, and these lungs are obtained from NIH, and I'll credit all the people involved in this at the end. And so what we did was we looked for in the lung of these COVID-19 autopsy samples, and what we found is the MLKL positive cells also have SARS-CoV-2, replicating what we saw in the cultures. However, when we look at caspase-3 and cells with the virus, we saw similar data that are similar to what I showed you before. So then what is the pathway? What is the mechanism? So we were able to really take a lot from this paper from Balachandra's lab at Emory, and they showed in influenza virus that virus can replicate, produce zRNA, which then activates ZBP1 to activate the MLKL phosphorylation. And so zRNA is a high-energy alternative to the right-handed aRNA that we typically think about, and it can be recognized by at least ZBP1 and also ADAR1. So we wanted to know, first of all, if SARS-CoV-2 infection can produce zRNA. And so we did zRNA staining with the antibody, and we were able to show quantitated here that indeed upon infection, we see increased zRNA production by SARS-CoV-2 infected cells indicated here. And next, we wanted to know if this is indeed zRNA signal or it's DNA signal or RNA signal. So we treated these samples with RNAs, and it got rid of all the zRNA signal. DNA's treatment did not get rid of any of the signal. So we believe this is attributed to zRNA. And I should say, credit where it's due, the Lee et al. Paper showed that SARS-CoV-2 can induce zRNA. And they showed that this is important for cytokine production. And we are looking at this in terms of signals in necroptosis. So we wanted to see next if zRNA that SARS-CoV-2 infection can induce ZBP1 expression, which is the receptor, innate immune receptor for zRNA in culture as well as in patients. And so we infected cells, and this is A549 cells, and this is HAE cells, and infected these, and we were able to see logarithmically increases in ZBP1, which is the receptor for zRNA, and we showed here by immunoblots. And in patients, we are also able to show that SARS-CoV-2 had higher amounts of ZBP1 protein. So now we have zRNA, we have ZBP1 protein. So are the 2 linked together? And so Kashin and Kat came up with this really cool procedure. The procedure is known by others, proximity ligation assay. What they did is really use this to ask if zRNA is close to ZBP1. So we have an antibody against zRNA, we have an antibody against ZBP1, and then using this PLA assay, which causes circular PCR amplification, we were able to really amplify the signal with a red tag, which is shown here, and you can see these red dots and SARS-CoV-2 infected cells. So this is looking at zRNAs that are bound by ZBP1. In mock infected, we really don't see any signals, and so by doing this, we can now link the two together in infected cells. And so we now think that ZBP1 is important. We knock out ZBP1 in HAE cultures, in the HAE cells, which are the human airway epithelial cells. And when we knock down ZBP1, indeed, MLKL is reduced. In fact, it's almost gone, indicating that these are linked. And when we look at caspase-3, it's very slightly modified. So we think this is the pathway and has not, in these cells, is not really linked to the apoptosis pathway. So then we wanted to look at different strains of SARS-CoV-2 virus. By that point, Delta and Omicron were really in circulation, and Delta is the more severe form, Omicron the less severe form. And what we found is Delta can cause a huge amount of cell death, more than Omicron, more activation of RIP kinase 1, RIP kinase 3, and ZBP1. So all the red ones are the Delta from the Delta infected cells. What happens in vivo? So our Duke colleague helped us with this experiment where we infected K18 with human ACE2 mice infected with Delta or Omicron, and we can show that the body weight loss is much more significant with Delta infected mice than Omicron infected mice, and the lethality is shown here. And then when we look at the PLA study of ZBP1 and zRNA, we showed that this pathway that we've been looking at also is heightened in Delta infected lung compared to Omicron infected lung. And this is quantitated here. And finally, we wanted to look at patients. So we were able to get several patients' samples from autopsy samples from NIH, and we look at those with very low viral titer to those with higher viral titer and show that there's a linear progression of those with higher titer with higher PLA of ZBP1 and zRNA. So we went all the way from cell culture to mice to human samples. So just to conclude, this is a way that we have used is using airway 3D cultures to look at SARS-CoV-2 infection in these airway cells, and they induce both necroptosis and apoptosis. But necroptosis is limited to infected cells, while apoptosis can be found in bystander cells, but and also infected cells. But only in bystander cells at low MOI do we see this process, which is apoptosis. And finally, we showed that the pathway is through ZBP1, recognition of zRNA, and we showed this all the way to patients. And finally, we showed that the more severe, which is probably the better replicating virus, the Delta virus, caused more robust cell death than Omicron. So it's depicted here with SARS-CoV-2 produced zRNA activating ZBP1, activating MLKL, and this is in primary cells. We believe in bystander. Some bystanders may go through apoptosis, but most of bystander cells go through this caspase-3 process, which probably led to gastromin E cleavage. So in the final example I'll give you is inflammasome as a mechanism for sex bias of periodontitis. Really, again, the majority of the credit really goes to Julie Marcheson, who's a long-term collaborator of our lab, who works in the dental school. And so the disease of periodontitis is really prevalent, but there is this sex bias in males. And so this is a major form of tooth loss. Like I said, there's a sex bias, but the mechanism is unknown. And so there's mild periodontitis, which can go to severe periodontitis, leading to tooth loss. So this is a really major problem. It's the sixth most common disease worldwide. It affects nearly half of American adults. And there's a prevalence shown in males, as I will show you in just a little bit. And the biology is really unknown. And here, this shows the total number of people with this disease, PD. And if you look at the distribution of males to females, there's a male bias indicated here. So Julie has set out really in her lab, we played a role in this to really understand what is the mechanism. So her lab has designed this simplified ligature-induced periodontitis model, where one puts a ligature that's 3D printed, indicated here, for each tooth, and therefore this part cannot be cleaned and causing gingivitis and as well as periodontitis. And then this is correlated with studies from humans and large study of 6,000 samples, and then also individual samples from getting the gingival tissues, as well as later samples of neutrophils, looking at male and female differences in single cell analysis. And Tomas is a student that Julie and I share, and John is a research associate in my lab who has really contributed significantly to our computational biology. And so here is this model again. It's a ligature induced, and one can look at the results of this. And what we found is increased IL-1 in the gingival upon activation with time, and this is just looking at different time frames. Very high amounts of IL-1 in these samples, and also increase of neutrophils, MPO1 positive myeloid cells, increase of osteoclasts, which would break down the bone, and increased drop in bone level. So all of these are shown in this mouse model. So the first line of investigation is to find if male mice also present with more severe bone loss and also more inflammatory conditions. So this is male mice and female mice and male mice looking at bone loss and looking at bone level in two different kinds of measurements. And what we saw is indeed female mice have bone loss, but the males have increased bone loss, replicating what one sees in humans. And this is also shown here. Remember, I just want to remind people that this is looking at very small, precise measurements, such as these, in terms of the loss of this difference in bone level. And in terms of looking at myeloid cells, we use MPO1. And in the females, there's an increase in MPO1, but then it drops. So it's not sustained, while in the males, it's sustained. And so there's increased levels of inflammation, again, mimicking what one sees in humans. And I should say this is unpublished work that is a collaboration between our two labs. And so to really address the question, if the inflammasome or IL-1 is involved, we put these mice through the periodontal model using wild type and IL-1 knockouts, females and males. In the males, there's a difference between IL-1 knockout and the wild type. So IL-1 exacerbates the disease. By contrast, in the female, we don't see this difference. Again, replicating what we are thinking about in terms of humans. In that human, I will show you later, the females have lower amounts of IL-1. So then they wanted to understand about therapy. So Pollyanna and Mustafa and Julie's lab started to do these experiments using a caspase-1 and 4 inhibitor called VX765 from Vertex. And they did an oral drug delivery, and then after that, look at bone loss in these. And what they found is the females actually didn't respond to this treatment. The males did. So upon treatment, they showed a reduced bone loss in the males. And it also inhibited many other processes that I will not show you, including bone resorption and osteoclast response in the males. And so looking at VX765 in more detail, we look at rank ligand, which is an important activator of bone loss, osteoclast. And what they found was, this is females, this is males. And if you just look at untreated, this is the amount of rank ligand in the females, and this is the amount in the males. The males have increased levels of rank ligand. However, if these are treated with the Vertex 765, the males have a reduction of rank ligand. In addition to that, we also looked at the amount of cathepsin K staining. This is important because this is a biomarker for osteoclast. And again, if you look at male females, so there's an increase, but the VX treatment, sorry, I want to move this so I can see it. Looking at cathepsin K positive osteoclast, positive signal is those with VX765 treatment. The females really didn't have a significant change upon VX765 treatment, but the males did, and there's a reduction in osteoclast. So in general, the inhibitors of caspase 1 and 11 was able to reduce bone loss, osteoclast number, and also MP01, which I did not show you. So with that, oops, we wanted to move into humans. And so looking at different human studies, one is just taking the gingival fluids, collecting these, and look for IL-1 differences. And these are very large numbers of human samples, and there's a difference between female and male. And I should say this is at the constitutive level. And then we also did a smaller scale study with existing analysis of existing data, looking at the cells in these fluid in terms of what are they. And what we found, the IL-1 expressing cells are clustered here in monocyte neutrophils, dendritic cells as expected, but also in this cluster, which are the epithelial cells indicated here. And so these are two main populations in the oral cavity that are expressing IL-1. And finally, looking at ex vivo, we took human neutrophils from males and females and looking at their response to IL-1 comparing IL-1 levels. So this is in the PBMC, so this is not in the gingival fluid, but looking at the response to LPS and N of 30. And what's really interesting is what we found is, again, looking at the females indicated here, the baseline, and those with compared to males, and this is with LPS treatment, males and females and males. The males have increased levels of IL-1 upon LPS treatment. If you look at TNF and other inflammatory cytokine, there's really no difference. So this is specific to IL-1. So in different cases, what we observe either in the gingival fluid or in the PBMC response, the males have increased IL-1 production, correlating this with increased periodontitis. And in mice, then we showed that when you knock out IL-1, there's a improvement in the disease in males, but not females. So basically what we're saying is when the condition is created where there's gingivitis leading eventually to periodontal stimulation, we see inflammasome activation, and this process seems to really have more profound effects in the males rather than the females. In the males, we see that if we block this process, we can block osteoclast and also in bone loss. So this may be one of the mechanisms to explain why males are more prone to periodontitis. So in summary, what I've done is really take you to two directions, and we have more directions in the lab. But these are two of the main directions. One is asking the question, what are innate immune receptors doing in other cells, that they have roles beyond innate immune cells? And this really stemmed from our very early finding when we were looking at NLRs. It turns out they are highly expressed in many reproductive organs. And we don't have any expertise to look at the reproductive system, but there are many very beautiful studies done genetically, for example, for NLRs, showing some NLR proteins are really important for the development of the embryo or even for just embryogenesis. So that's another important field that we have not touched on. Instead, we have focused on these in innate immunity and found they really a plethora of roles of innate immune receptors in adaptive immunity. In the second line, we're really trying to ask the question, how do we translate what we know into human immunology? And I gave you three examples. One is human 3D cultures that we use extensively for SARS-CoV-2. The other is genetically modified humanized animal models, where we replace the entire gene and its regulatory elements from replace the mouse gene with the human counterpart. And finally, doing primary human studies and validating these with mouse studies, which can validate which reproduce some of the features in the human, including sex bias. So I just want to thank you all very much for your attention, especially acknowledging the important people in the lab. So the adaptive immune studies, I mentioned Emily and also Cece, and we couldn't have done any of this without help from our colleague here, Isang, in the AIM-2 studies, and Yulia with the STING study. And these are some of our previous members who participated, and these specifically are members who worked in the T cell or B cell components. And then our SARS-CoV-2 study was done by Kat initially, and Kai-Shin, and crediting Dinka for leading our BSL-3 effort in the beginning, and then the two of them going to Duke all the time to do these experiments, which is 12 miles away from us. And then thanking our collaborators at UNC, which allowed us to do all the airway cell culture, including Scott and Richard, and then colleagues at NIH, at Duke, University of Michigan, WashU, and Michigan State, and then some of our other colleagues who contributed mice or samples. And then the studies that we did with periodontitis are done in collaboration with Julia. Like I said, she really deserves the giant share of the credit. And Tomas is a shared student, and these are some of her lab members. And Karen is a former postdoc, but she's now a full-time collaborator with Julie, and she has her own lab. And these are other people from my lab who have helped with the project. And finally, a conflict of interest. I'm a co-founder of Invention Therapeutics, and I just want to thank you very much for your attention.
Well, thank you very much, Jenny, for this wonderful lecture. Just to remind the audience that you can ask Jenny questions through the Global ImmunoTalks Twitter account. The instructions are on your screen now, and they will also be on the chat. And Jenny will endeavor to answer your questions over the next couple of weeks as they come in. So it just remains for me to close this week's session. Thank you all very much for tuning in, and don't forget to tune in again next week where we will have a lecture delivered by Manna Clapworthy. And so Jenny, once again, thanks for participating in Global ImmunoTalks, and see you all next time.
Thank you so much.